Overview of bacterial phenotypic identification based on cultural and biochemical characteristics, 1 Professor Mokhtar Kamara. Identification of bacteria colonies by diagnostic laboratories is based on phenotypic characteristics. Heavy checkmark cellular morphology, usually shown by gram, bacteria. Heavy checkmark colonial morphology. Heavy checkmark biochemical metabolism. Heavy checkmark genome identification, molecular methods. Heavy checkmark spectral profile of bacterial proteins, MALDI-TOF MS. 2. Bacterial colonies of a single species when grown on specific. Morphology, heavy checkmark characteristic size, heavy checkmark shape, heavy checkmark color, heavy checkmark consistency, heavy checkmark pigmentation. The growth rate of certain organisms are variable depending on 3. Colonial shape is determined by the edge and thickness of the colony. The edge may be smooth, entire, or irregular and serrated. If the colony is thicker in the center than the edge, it is said to be raised, or it may be relatively uniform, appearing like a disc. The texture of the colony is also important. It may vary from dry and friable, easily crumbled, to butyrous, buttery, to sticky, and the surface may be smooth, wet, dry, or granular. Some organisms produce a pigmented colony, which can aid in the identification process. For example, Pseudomonas aeruginosa, Serratia marcescens, Staphylococcus aureus. Other microorganisms produce hemolytic colonies. Group B Streptococcus, Streptococcus pneumoniae, S. aureus. 4. The size of bacterial colonies is generally uniform within a species. Streptococcus species are small, usually 1 mm in diameter. Enterobacteriaceae are larger and usually 2 to 3 mm in diameter. Bacillus species are much larger in size and usually 2 to 7 mm in diameter. 5. Appearance of colonies. Smooth colonies S. Smooth, smooth, shiny, and moist with 2 to 4 mm in diameter. Except Yersinia, smaller colonies. Rough colonies are rough, dry, irregular contours, and matte in color. Mucus colonies, M, with diameter 10 mm, tend to confluence, e.g. Klebsiella large. Fatty and shiny colonies, dwarf colonies, strains deficient in their metabolic chains, urinary E. coli strains, special case, proteus, agar invasion, uniform matte. 6. Smooth bacterial colonies, 7 Staphylococcus aureus colonies. Rough bacterial colonies, 8 Pseudomonas aeruginosa colonies, Bacillus colonies. Mucus bacterial colonies, 9 Klebsiella colonies. Swarming colonies, 10 swarming proteus culture in blood agar culture of proteus on nutrient agar medium. Colony pigmentation, 11 non-diffusible pigment Staphylococcus aureus versus white Staphylococcus spp. Colony pigmentation, 12 Escherichia coli EMB culture colonies. Colony pigmentation, 13 brick red pigment of Serratia marcescens non-diffusible pigment. Colony pigmentation, 14 chromogenic culture media. Colony pigmentation, 15 diffusible pigments Pseudomonas aeruginosa versus Pseudomonas fluorescent. Type of hemolysis, complete hemolysis versus partial hemolysis, greenish, 16. Type of hemolysis, 17 CAM test, Christie, Atkins, E.T. Munch-Peterson. Demonstration of a secreted cytolysine which causes the formation of pores in the membrane of sheep red blood cells previously sensitized by sphingomyelinase secreted by certain strains of Staphylococcus aureus S. agalactiae. Butterfly wing image Staphylococcus aureus stria Streptococcus agalactiae stria GBS gas S. aureus. Identification of bacteria into genus and species respiratory metabolism, oxidase, catalase, protein metabolism. Presence of urease, indole production by degradation of tryptophan. Presence of enzymes, decarboxylases, deaminases, coagulase, DNase, Lipid metabolism, lipase, glucidic metabolism. Fermentation of sugars, glucose, lactose, mannitol, maltose, sucrose, etc. Use of citrate as unique carbon source, Simmons citrate, Christensen citrate, hydrogen sulfide production, gas production. 18. Catalase test 19. 20. Indole production 21. Positive versus negative. Negative versus positive. 22. Decarboxylase production 23. 24. S. Aureus coagulase etDNase production test to coagulase positive DNase test. 26. 27. Citrate utilization 28. Nitrate reduction 29. Gram-positive coccus API Galleria 30 API 32 strep API 32 staph. API Galleria Enterobacteriaceae 31. 32. 33.